Welcome to this lecture on microbial selection within environmental biotechnology course. Today we emphasize the use of selective enrichment of desired bacteria. Environmental biotechnology is all about selecting the right microbe for an engineering process. To do this, we need to understand what is microbiologically possible. How do microbes function and compete with each other? And most importantly, how can we engineer the conditions to give the desired bacteria a competitive advantage? To begin, we will go through the history of microbiology in Delft, which laid the foundation for the field. Delft has a rich microbiology legacy, starting over 300 years ago with Antoni van Leeuwenhoek. Using his groundbreaking microscopes, he was the first to observe bacteria, describing them as small animals. He saw them in the water from Delft's channels. His detailed drawings revealed a hidden microbial world invisible to the naked eye. At Delft University of Technology, Professor Beyerink advanced microbiology by moving beyond mere observations. He initiated the use of selective enrichment media to culture and isolate specific bacteria. One of Beyerink's students, Baas Becking, refined this idea with his famous quote, "Everything is everywhere, but the environment selects." This means microbes are ubiquitous, but the environment determines which ones thrive. In environmental biotechnology, we engineer conditions to select the microbes that execute the conversions we want to use. Professor Kluiver, Beyerink's successor, introduced the unity of life concept. All living organisms, bacteria, plants, animals, share similar growth processes. The biochemical pathways for producing the molecules needed to build a cell, termed anabolism, are universal. The microbial diversity mainly originates in the diversity of energy generation, termed catabolism, driven by redox reactions using electron acceptors like oxygen, sulfate or nitrate. Microbial selection depends on the environment, often more than the microbe itself. This shifts our focus to microbial communities, how they compete and which ones dominate under specific conditions. While anabolism, building the cell material, is similar across organisms, it is the catabolism, the energy harvesting, that is the major selective factor. This process is called microbial community engineering. It involves designing culture conditions to promote specific microbial conversions. Industrial biotechnology starts with a very well-known organism. This can be genetically engineered and is cultivated in a standard completely mixed reactor. Environmental biotechnology, however, starts with a microbial community. We engineer the reactor conditions to favor the desired microbe, allowing it to dominate and perform the target function. Microbial community engineering is traditionally widely used in wastewater treatment for processes like biological phosphate removal, nitrogen removal and biogas production. In summary, environmental biotechnology exploits the potential within microbial diversity and ecology to design processes that convert waste into harmless compounds or valuable products. The key is creating the optimal conditions in selecting the desired microbe. This course will dive into microbial selection principles and explore innovative technologies built on them, such as the Anamox processes, aerobic granular sludge and bioplastic production. Get ready to discover these cutting-edge solutions.